Welcome back. And in this lesson, we are going to learn about BioSwift biosets. And biosets allows us to hack sequences that are unique and eliminate redundancy. So let's create a BioSwift bioset. And this time, let's use our diabetes datasets. Let's start with the nucleobases. So I'm going to say biogen D to pull up our BioSwift diabetic sequence dataset. So now let's do a count on the sequences in this diabetes dataset. And this is showing 10 different sequences in the dataset that are in fact unique. If you were to count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. It's only counting 10 because there are 10 unique sequences. So it is somewhat indubitable that two of them are the same. Now, I had made a mistake in saying that we were going to initially pull the nucleobase versus the sequence. So let's go ahead and pull the nucleobase now. And that would be the FASTA file for diabetics. So, and there we have that file. And we'll do a count on that. And that is showing 4 unique nucleobases in that particular data set. We have adenine, cytosine, guanine, and thymine. 4 nucleobases. Because BioSwift biosets only allow unique sequences and or nucleobases, eliminating redundancy. I could add 4 uracils, and that would show me 5 nucleobases, even though I added 4 uracils. And as you can see, it shows me 5. Five nucleobases. I could print them out and they would show the 5 nucleobases. As you can see, TACGU, I could sort them. And here you will see that they were sorted. I could print it so you can see it a little bit better. And you can see that adenine, cytosine, guanine, thymine, and uracil is in fact in ascending order. I can also remove uracil from that particular sequence. And if I were to print it out, you would see that uracil is no longer a part of that sequence, changing it from RNA back to DNA. Next, I'm going to create another variable for a sequence, and we will use the cancer data set this time. I think I'll use the sequences. And I'll create a second sequence. And we'll use the same, but I'll take this one and I'll make it our uracil there, and I'll make it a uracil there. Okay. So now we have two sequences to compare. So I'm going to say let DNA set equal sequence 1 dot union sequence 2. And this prints 6 different sequences. So let's find out if we have 5 sequences in the first sequence and we have 5 in sequence 2, but when we concatenate the 2, we have 7. So there's 3 sequences that are redundant. We use intersection to find out redundant sequences, like so. So considering that it's a 0 index, it was actually only 2 sequences that were redundant in the 10, or what would be 10. That is the sequence that starts with CTGA and the other sequence with a prefix of 4 thymines. So CTG here and here, and 4 thymines here and here. Those 2 were eliminated from the concatenation in the BioSwift set. So our final BioSwift bioset function that I want to share with you is the subtracting function. And what that does is when you are comparing 2 sequences, it will take any sequence from the first set and any matching sequence from the second set, it will remove ultimately and create a new set, removing the redundant sequences. And as you can see, it eliminates any redundant sequences and creates a new set with unique sequences. So that is going to conclude this lesson on hacking with biosets. In our next lesson, we are going to work with BioSwift dictionaries. See you there.